So we'll start with the labeling part. On the first picture, I didn't ask you to label everything that you could have labeled. So I'm going to name everything that you should realistically know how to label, but you only have to give yourself marks for the ones I asked for. Part A, since it's showing the blood coming away from the heart towards the kidney, Part A is an artery. Now, since I pointed to the piece of the artery that isn't yet branching off, it's technically not the renal artery yet. This big piece of artery is your aorta. When it gets over here, that's when it's your renal artery. Now, if you recognize that it was an artery, that would be my main point of interest right now. When we start talking about blood in one of our next sections, then we'll really get into specifics about arteries and veins. But the idea is that you recognize that blood is coming from your heart towards your kidney. And when it branches off, then it's called the renal artery. B is a vein. Specifically, it is your vena cava because it's not the branch. The branch down here is your renal vein. I'm looking for the idea of vein. If you recognize that it was a vein, that's what I'm looking for on this particular one. Does anyone have any questions about those blood vessels there? The reason I am harping on blood vessels, even though we're not doing blood, is because that is what is getting filtered in your kidney. And so it's important for you to recognize if the blood is coming in or coming out of your kidney, because it will have different levels. Now, C and D are really close in name, and it is really easy to mix them up. That's why I always ask them. C is your ureter, and D is your urethra. See how D has one E and C has two? You have two ureters and one urethra. That is always how I remembered which one was which. Now, just so that we're keeping things complete, don't forget that this is your kidney and this is your bladder. Now, does anyone have any questions about anything on that first picture? So then the second picture, which is obviously your kidney but blown up a little bit, E is the renal cortex. If you just said cortex, I would accept that. If you just said renal, I wouldn't. F is the renal pelvis. And this little triangle-shaped thing, even though I didn't ask for it, is the renal medulla. So those are the three layers, let's say, of kidney tissue. G, because it's just the whole structure, I was only looking for you to call it a nephron. And very likely the next time we do a daily review, I would ask you more specific things about the parts of a nephron. Now, does anyone have any questions about A, B, C, D, E, F, or G there? So that first part is out of 7. Now, for the second part, even though I asked you 2 questions, we're only going to make it out of 1 because I want to focus on the idea of waste. We'll get into the idea of oxygen a lot more when we talk about respiration and when we talk about blood vessels. Plus, I realized as I explained this question, I said something that was not true, so that was mean of me. The idea of waste is really what we're focusing on with your kidney. Blood that comes into your kidney should have lots of waste in it. Then your kidney filters the blood, and the blood that comes out has less waste. So, where should there be more waste? There should be more waste in A, the artery, because it has not yet been filtered. So if you, for example, labeled B as the artery, but then you said B was your answer to this, I might end up giving you part of a mark there because you said the right blood vessel. But the artery should have more waste. That's the blood that's coming in. It has to get filtered. Now here's the part where I said something incorrect right at the beginning. Your renal artery also has more oxygen. So the answer is A for both of them. So since you answered both questions, give yourself a mark for whichever one you said A for, because you're right, and we'll make it just out of one so that you don't get penalized because I said something ridiculous at the beginning. Before we can talk about how your kidney filters waste, we need to talk about where the waste comes from. So you need to understand this process of deamination. So it's got a couple of parts to the words. D means like I'm undoing it, I'm taking it away. Amination, doesn't that kind of sound like an amino acid? That's what's happening. I am undoing an amino acid. That's what deamination is. So the official definition is the breakdown of amino acids. And I'll just remind you that an amino acid is the building block of a protein. So if you consume a lot of protein, your body will have to do more of this deamination process. For example, if you consume protein shakes and your body is not using all of the protein to build muscle, then you will have a lot more of this process happening. And so I have heard someone refer to it before as the protein powder pee, where your pee is really yellow and maybe really strong smelling. This is why. So this happens in your liver. Now your liver has shown up on a number of occasions. Your liver has to do with digestion. It produces bile. Your liver has to do with excretion. It does this process of deamination. We alluded to the fact in chapter 6 that your liver does other things too. It helps with blood sugar. It helps with red blood cells. I would ask a question about your liver because it does so many things on like a final exam type scenario. So, what happens during deamination, which is occurring in your liver? AAs, which is my abbreviation for amino acids, although I am aware it means something else, are broken down into two groups. Now I'm using the correct scientific names here, and since this isn't Chemistry 30, I wouldn't expect that you can define these for me, so I'm going to give you some shortcuts. One side of the amino acid is the amine group. It has nitrogen in it. The other side of the amino acid has some C's and O and H in it. That's the carboxylic acid side. And so what we're saying is we're taking this amino acid apart. Now the side that we're calling a carboxylic acid, carb because there's carbon in it, oxalic because there's oxygen in it, acid because there's H in it, so there's a reason for all of those things in the name. One side of it can be used in cellular respiration. Now I'm going to show you a picture on the next slide of exactly how that would happen. This would be a fantastic link between this chapter on excretion and the chapter on cellular respiration because this is all happening in your liver. Now the part that interests us a little bit more immediately is the amine group. That's the part that has N in it, the nitrogen. Once this part gets chopped off the amino acid, I combine it with another hydrogen to make ammonia. So I have a little picture here to illustrate this. On an amino acid, there's a piece on the side that has hydrogen and nitrogen, and a piece on the other side that has C and O and H. That's the amine group and the carboxylic acid group. Basically, I'm cutting this piece off. So I'm taking the part that has nitrogen, it sticks out on a little branch on the side, and I'm cutting it off. I wouldn't expect that you can just draw from scratch an amino acid, but maybe if I showed you a picture of it, could you identify that it's the part with nitrogen that I'm cutting off? Now, since there's only 2 hydrogens, I have to add another hydrogen atom so that I can get ammonia, which is NH3. Again, drawing chemical structures is not the purpose here. Understanding that it's the part with nitrogen that we're focusing on is. Now, ammonia is pretty toxic. That's why you shouldn't drink Windex or household ammonia, so please don't. So ammonia is toxic. So your body does not want to keep ammonia around if it is possible. So it should get converted to something that is less toxic. And that compound is called urea. And urea I get by adding CO2, carbon dioxide. Then I can take this compound urea and take several molecules and combine them to make uric acid. So I have 3 compounds. Ammonia, which realistically should not be hanging around for very long. Urea, which I got by adding CO2. And uric acid, which I got by putting a few molecules of urea together. The liver then takes these 2 compounds, urea and uric acid, and puts them into your blood. Your blood takes these compounds to your kidney, and your kidneys filter the waste. So at the bottom here, I have a little picture so I can make you a little summary of the compounds so that you can maybe see it in a more visual way instead of a text-based way. So there's my amino acid. I'm taking the part with N and I'm cutting it off to get my ammonia. Then I add CO2 and I get urea. Then I take at least 2 of them and I get uric acid. So that would be a summed up version of what that second paragraph is saying. I get ammonia, I add CO2, I get urea. I take at least 2 of them, I get uric acid. Now, would anyone like to ask any questions about this process that's happening in your liver? So these are our waste products, the main ones that we're talking about. And I'm highlighting them so you know what you should be looking for in terms of waste. These are the types of molecules we would discuss. Now the next slide has a picture just to show you that bit I mentioned before about cellular respiration. I don't expect that you would draw this whole picture. I just wanted to illustrate how I could take a piece of an amino acid and use it in cell respiration. So I'm just going to highlight here, there's deamination happening. Because of deamination, I will eventually get a molecule of glucose, and glucose will go to your muscles and go do cellular respiration. Now there's a lot of chemical things that happen in between there, but the basic idea is that because of deamination, I will get a molecule of glucose. There'll be a couple of steps in between. Once I have that glucose, I can go do cell respiration. Because we're talking about waste, and we're focusing on the wastes that animals produce, it would be different in a plant. We're focusing on human physiology, so we're focusing on the kind of things that animals make. I want you to be aware that your kidneys are one of several organs that produce waste. In this chapter, we're focusing on your kidneys, but that's not the only place that waste comes from. For example, your lungs produce CO2 and water. Every time you breathe, CO2 comes out, a little bit of water comes out. Your skin produces waste. If you imagine the idea of sweat, every time you sweat, you are producing waste. And I want you to look at what your skin produces and compare that to what your kidneys produce, and notice how similar it is. Water comes out in both of them. So does urea. Now this thing where it says salts. Salt is a very broad word used in chemistry. A salt is usually some part of an ionic compound. And we're not going to be incredibly specific about all of the possibilities there. Just know that when we say salt, we don't just mean table salt. We mean ions, positively and negatively charged ions that come out with waste. So notice your skin and your kidneys are producing the same stuff. So when you sweat, what comes out of your pores is very similar to what comes out in your urine. Hence why sweat smells so bad. It's basically the same thing as pee. Now your intestines you already know about because we discussed that already. Undigested material comes out. And so does bile. Bile is what gives your feces the characteristic color that it has for the most part. And so keep in mind that we are focusing on metabolic waste here. And metabolic waste is waste that comes from a chemical reaction happening. So what's happening in your kidneys is different than what's happening in your intestines. Your intestines are producing waste that you didn't digest. It's stuff that you didn't digest, you didn't do a chemical reaction with. Your kidneys and your skin are producing waste from a chemical reaction. The reaction is deamination. Yes, so what I'm saying is pee in your hot tub. Basically, that's what I'm saying. Basically. So this next little chart is to help illustrate some differences between organisms that live on land and organisms that live in water. So on this topic of peeing or not peeing in your swimming pool / hot tub, if you've swam in a lake, you have swam in pee because fish live there and they pee in it. And so I've got a little comparison here. I'm going to make a short version of this so you don't have to necessarily draw all of this. But I'm going to compare a freshwater fish, a marine fish, and a terrestrial vertebrate, which is what we are. We are vertebrates. We have a spinal cord and column, and we live on land. Now, freshwater fish are the ones that would live in lakes. Bony marine fish are the ones that would live in ocean. And I'm making a comparison because you might be aware ocean water is saltier than lake water is. So if I was to compare what is happening in each of these scenarios, just so that you understand why maybe urine looks different or is different, I'm going to focus on these test tubes. So if I look at the test tube for the freshwater fish, it's pretty full, but the color is really pale. So a fish that lives in fresh water produces a large volume, but the urine is not very concentrated. That's basically the rule of thumb with urine. If lots of it is being produced, it's probably not very concentrated. If a small amount is being produced, it's probably very concentrated. Since these fish live in water, they can afford to waste water. That's basically the idea. They live in water, water is always available, and so they are not super worried about conserving a whole bunch of water. The terrestrial vertebrate, if we look at its test tube, is less full, but the color is much darker. So the volume is moderate, but the urine is more concentrated. And that's because we don't have water available 100% of the time. Now humans have it a little bit better. We can access water even if we couldn't find it ourselves in the wild. But for other terrestrial organisms, water is not always available 100% of the time, so we're concerned with conserving it. We do not want a lot of water to leave with urine. The bony marine fish lives in salt water, and so salt is constantly coming in and out. And salt is a big factor that determines whether or not water is absorbed or not. If we take a look, their urine is very, very small in amount. And it's concentrated, not quite as concentrated as a human's, slightly less. So if I was going to compare these three organisms, I would ask you something like, is the volume high, medium, or low? That's lakes, humans, oceans. And what's the concentration like? Is the concentration low, like in a lake, high, like in a human, or just a little bit less concentrated? So that ocean one would be the medium concentration. All right, so we are going to talk about what is happening in the nephron. So you already have a picture of a nephron, so I'm just repeating with a few different looking diagrams what's going on. But we're going to start by talking about filtration. The first thing that happens in the nephron is that your blood is filtered. We sometimes call it force filtration. That's because your blood is coming through an artery. And arteries, like we said on the first day, have high pressure. So blood is getting pushed in, pushed through. And it's going through the glomerulus. So we labeled that before. It's this little ball of blood vessels that kind of looks like a crisscross, like it has a grid or a grate on it. And your blood is getting pushed through there. Approximately 20% of your blood goes through that filter and ends up inside Bowman's capsule, which is the very first piece of your nephron. So if I look at this, that is where my nephron starts, Bowman's capsule. The things that are coming into it are all blood vessels, and they're part of the process, but the actual nephron starts with that piece. Now, I'm going to give you some average numbers here. But what I really want you to focus on is this 20%. I could give you a different volume and ask you how much of that blood will end up inside of the nephron. So for an average adult human, about 600 milliliters of blood flows through the kidney each minute. That means that 120 milliliters of fluid is forced into Bowman's capsule. Now, how did I get from 600 to 120? I multiplied by 0.20. That's the decimal for 20%. Some of you might know how to use, because I'm not good at it, use the decimal number. You could also say 600 times 20%, I think, on most calculators and get the same thing, but I don't know if the touch sequence is the same. Now, this is a pretty easy calculation. You could do this without a calculator, find 20% of something, but you would be welcome to use your calculator if I ever asked you a question. Now, here are the things that should. Red blood cells should stay in your blood to filter. Proteins should go through the filter. They are too big. Platelets, which are another type of blood cell, should not get. So I've got a little picture here. Little gray lines showing kind of a grid are supposed to represent So reabsorption is where your body is going to take back anything that got through the filter that it actually wants to keep. So some of the things that go through, because they're small enough to fit, are not things that your body wants to get rid of. So out of the 120 milliliters on average that got into the nephron, only about 1 milliliter of that will form urine. That is less than 1%. It's a decimal number. We're not going to get too hung up on the details here, but it's less than 1%. And this is what should be happening normally. Most of the time, your blood gets filtered, and then your blood takes back all of the things that it would like to keep. So how does this reabsorption happen? Well, there are 3 steps. The first step is active transport. Now, active transport, so that everyone is aware, uses ATP. That's our cellular energy. We know that from our first unit. So this part requires energy. It can't happen without energy. The idea with active transport then is that molecules that won't move because of diffusion, they won't move just because of the concentration gradient, are moved purposefully. So sodium ions, which would be abbreviated Na positive, glucose, which I could write as C6H12O6, or amino acids, which have a variety of formulas, so I won't write them all down. They all have nitrogen in them, are active transported from the filtrate into the blood. So I'm going to highlight this part. Everything on this page is moving from the filtrate, what got out of your blood and into the nephron, back into your blood. And this direction would become important because the thing we'll talk about on the next slide is the other way around. Now, because they're being actively transported, we actually know something else about them. In active transport, things are always moved to an area where they're already in high concentration. Diffusion, which is passive, would be the opposite. So I'm just going to make a note here. You would have learned in grade 10 science that in active transport, molecules move from low to high concentration. So they're not trying to spread out. We're trying to get them all on one side of a membrane, let's say. In this case, we're trying to get all of the sodium we can and all of the glucose we can out of the filtrate back into the blood. Now, the second thing that happens is charge attraction, and this is passive. That means it happens naturally. We don't need to use any more cellular energy. And the idea is that things that are negative, chloride ions and bicarbonate ions, which could be abbreviated like that. This is CO3. Since I already active transported sodium, which is positive, these things that are negative are attracted to things that are positive, and they follow naturally without extra energy required. The third thing that's happening is water reabsorption. This is also a passive process. Now I'm going to put some words here. Well, there's some words here. Hypertonic and hypotonic. You might recall learning, again, in Science 10. Hyper means more. Hypo means less. Since I'm using the words more and less, these terms are always relative. I have to compare two things to be able to use them. I couldn't just say, your blood is hypertonic. Hypertonic compared to what? So if I compare your blood and the filtrate, since all of these things just left the filtrate and went back into your blood, now your blood is more concentrated. So tonic means concentration, essentially. So we're saying your blood is more concentrated and the filtrate is less concentrated. The gradient, which means the difference between your blood and the filtrate and their concentrations, is highest at the tip of the loop of Henle, which is the part of the nephron that dips down into the medulla. So I've got a picture here. Here's your loop of Henle. And right at the tip, right at the bottom, that's where the difference is the highest. So that's where most of this process is happening. Water will move passively by diffusion, which for water is called osmosis, from the filtrate into the blood. And the reason water will do this is because water is on this permanent quest to equalize concentration differences. So water's goal is to move to wherever the concentration is higher, essentially to water it down. Now, where is all of this happening? 3 spots, really. The PCT, the proximal convoluted tubule, the loop of Henle, and the distal convoluted tubule, the DCT. So first, in the glomerulus, blood is filtered. In the next 3 parts, I'm doing a ton of reabsorption. I'm taking things back from the filtrate towards the blood. Now, if you're interested, in your textbook on page 312 and 313 are some diagrams, much like the one I have here, that shows things going in and out so that you could refer back to it. Does anyone have any questions they'd like to ask about reabsorption? So then the last thing that we have to talk about today is secretion. This is the third thing that happens in the nephron. First of all of secretion is active. That means it uses ATP, and molecules are always moving from an area where they're already low in concentration to an area where they're already high in concentration. So we're forcing molecules to move against their natural process of diffusion. Everything in the secretion section is going from the blood to the filtrate. So this is the part where your body is adjusting and taking every waste product it can find and forcing it into the nephron. Some of them will be in there already because they got filtered in, but your body is going to adjust so that all of them get in there. So the idea is that we're getting rid of waste. That's the purpose of secretion. Some of it will already have been filtered, but some of it won't have got through. Remember, only 20% goes through. So the wastes that we're going to actively transport from the blood to the filtrate are the products of deamination. There are 3 of them. I just mentioned them earlier today. Ammonia, urea, and uric acid. Now remember that there should be very little ammonia kicking around because it's toxic. And I'm mentioning it only because it's one of the products of deamination. And then I listed a few other substances that usually are actively transported from the blood to the filtrate. Histamines is a substance that your blood produces when you're trying to combat an infection or you have an immune response. And anything that's in excess. So this last point here is really about excess. If you have too many hydrogen ions, and we'll talk about this again because it affects pH, if you have too many of a particular type of mineral, like too much calcium or too much iron, this could be the spot for it. If you have an excess of a particular type of drug, this would be the place that it is secreted so that we can get rid of the excess. Now, if I take a look at the nephron, you'll notice that in both of these spots, so this is the PCT and the DCT, there are some things that are going towards the filtrate. There's an arrow pointing from where the blood vessels are towards the nephron. Secretion happens mostly in those convoluted tubules. The loop of Henle is almost exclusively for water reabsorption. So that's where those things are happening for the most part.